So hello everyone, Assalamu alaikum warahmatullah. Today we are going to speak about the galactose and the galactose pathway and which enzyme they are used in this pathway, and if this enzyme they are deficient, what would we would have, like which disease would develop. So let's begin. So the galactose, from where do we get the galactose? The galactose, it's mainly come from the lactose, and the lactose it's found in the milk, the dairy products, and so on. So once we drink a milk, so we would get lactose inside our body. Then the lactose, it will be broken down into glucose and galactose by the lactase, and this enzyme is found, like this occur in the brush border of the small intestine. So once we have the lactose converted or will be broken down into glucose galactose using the lactase. So we will have the lactase right now. So what would have develop, or like what is going to do, what is going to happen after that? So once we will have the galactose, it can go through two pathway. The first one is galactose 1 phosphate, the second one galacticol. Let's start by the galacticol. So they are going to use the aldose reductase enzyme, which is going to convert the galactose into the galacticol via aldose reductase, and they will not be further metabolized. It will stop here, the galacticol. Unlike the galactose 1 phosphate, so once it uses the galactokinase to convert like the galactose into the galactose 1 phosphate, there would be the galactokinase. They would use also ATP to be converted to the ADP. And if you would see an enzyme contain kinase in it, so that would mean it's going to add a phosphate group in it. For example, as we can see here, we have the galactose. It would become galactose 1 phosphate. The 1 phosphate, it comes from the kinase because it's adding a phosphate group. So once we would have the galactose 1 phosphate, it will be converted to the glucose 1 phosphate via uridyl transferase, or it's also called the galactose 1 phosphate uridyl transferase. And they are going to use the UDP glucose converted to the UDP galactose. And if we want to convert it back to the UDP glucose, we would use the 4-epimerase. So by that we would have the glucose 1 phosphate, which can be used further in glycolysis or glycogenesis. So let's repeat one more time again. We said if we drink a milk, we would have the lactose. Lactose will be broken down to glucose, galactose, by the lactase, which occur in the brush border of the small intestine. And then the galactose, they can go through two pathway. There would be the galacticol that can be converted to the galacticol using the aldose reductase, and the second one by the galactokinase converted to the galactose 1 phosphate, and then the galactose 1 phosphate, so it can be converted to the glucose 1 phosphate. Then it will be used in glycolysis and glycogenesis. So I have like an image we can use here. So as we can see here, we have the galactose converted to the galactose 1 phosphate using galactokinase, galactose 1 phosphate to the glucose 1 phosphate using the galactose 1 phosphate uridyl transferase. And as I said previously, if there's a kinase, that means they are going to add a phosphate group. So for that reason, we have a phosphate here, and we are having phosphate. Once we have the glucose 1 phosphate, as we can see here, it can be used in a glycogenesis, or it can be used in a glycolysis in this pathway. So this is in regard for the galactose and how it is like its pathway. So now if we would have an enzyme deficiency in any of these enzymes, let's start by the galactokinase deficiency. So deficiency of the enzyme. So if there is a galactokinase deficiency, what would happen? So as we can see here, that galactose, they will be converted to the galactose 1 phosphate using the galactokinase. This disease, or like it is an autosomal, it's an autosomal recessive. So as we can see here, like which enzyme is deficient is the galactokinase. So we cannot convert the galactose into galactose 1 phosphate, as we can see here. Instead of that, the galactose, it will be shunted to the next pathway, or to the second pathway, which is to the galacticol, using which enzyme? Aldose reductase. So if our diet have galactose inside it, so galacticol is going to be accumulated there, and the galacticol, it's toxic to our body. Why it's toxic to our body? Because as we said previously, that galacticol, it will not be metabolized further, like it will be staying like that, so it will be accumulated. And why it's toxic? The reason is galacticol is osmotically active. So what does that mean, that it's osmotically active? That means that it's going to draw water into the cell. So because we would have galactokinase deficiency, so it will be shunted from the galactose so that means that it will be shunted the galactose to the aldose reductase, which means that aldose reductase is going to convert galactose into galacticol. So this enzyme, as we can see here, is deficient. It cannot be forming, it cannot form the galactose 1-phosphate. So galacticol is going to accumulate, and as I said previously, that galacticol, it is toxic. Why it's toxic? It's osmotically active. It's going to draw water inside the cell. So once you would have water inside the cell, especially in which tissue they are going to be active, especially in the tissue which have the aldose reductase is active there. For example, we have the lens of the eye. So that can lead to cell swelling, that can lead to structural damage, that can lead to dysfunction. So due to the galacticol accumulation, all of this is going to occur. For example, you have the lens. As I said previously, that the lens, it is aldose reductase is active there. So the infant or the patient will develop cataract. Due to what? Due to the water is being influxed inside the lens, then the lens fiber is going to swell, resulting in lens opacification, leading to cataract. So what are the symptoms that they are going to develop in an infant who have galactokinase deficiency? So before we dig inside the symptoms, it's relatively benign and mild condition in the case of the galactokinase deficiency. So here we have a mnemonic, which is galactokinase. To remember, like the enzyme is that is deficient, which is galactokinase deficiency. So let's remember the first 5 letters from this enzyme. Let's start by the G. We have a galactose. They would appear in the blood and in the urine, which means galactosemia and galactosuria. So we will have the infant. They will present with galactosemia and galactosuria. There would be abnormal eye tracking. There would be lack of social smile. It's an autosomal recessive disease, and they would have infantile cataracts. We said that the cataract they would develop due to what? Due to the galacticol accumulation, resulting in the lens fiber swelling, lens opacification, and cataract. This is for regard the galactokinase deficiency. Now we have the second disease, which it is called the classic galactosemia. In this disease, the deficient enzyme is the uridylyl transferase, as we can see here. So the galactose 1-phosphate, they cannot form the glucose 1-phosphate via this enzyme because it is deficient, as I said. So the galactose, it would accumulate, galactose 1-phosphate, it's going to accumulate. In addition to galactose 1-phosphate, we would have the galacticol, it would be accumulate also, because as we said, like the galactose 1-phosphate is deficient, so it will be shunted again to form galacticol. So what are the symptoms that they are going to develop? These symptoms, they will start when the infant is fed formula or breast milk. Why breast milk? Because we said the lactose, it comes from what? From the milk. And so we would have the lactose will be broken down into glucose and galactose, and we have the galactose again. So what are the symptoms that they will occur? So we have a mnemonic here, which we would have the classic, same name to the disease, classic galactosemia. So what are the classic? For the C, we have the cataract, severe infantile cataract. For the L, we have liver enlargement, hepatomegaly. For the A, we would have autosomal recessive. It's an autosomal recessive disease similar to the galactokinase deficiency. For the S, we will have sepsis. For the second S, start with feeding, as I said previously, nausea, vomiting, diarrhea after milk. And we would have intellectual disability and color yellow and jaundice, failure to drive. So, like I have a comparison here. We would have in the case of the classic galactosemia, there's hepatomegaly, there's sepsis, there's intellectual disability, there's jaundice, there's failure to thrive. Unlike galactokinase deficiency, that there would be only infantile cataract. What's the reason behind all of that? The reason is the source of toxicity. Like in both disorders, galacticol, they are going to build up, resulting in cataract. While in the case of the classic galactosemia, there's an additional toxin, which is the galactose 1-phosphate. So this galactose 1-phosphate, what it's going to do? Because it's phosphorylated, and as I said previously, that we would have the galactokinase, and it's active here in the case of the classic galactosemia. And the kinase mean that it's going to add a phosphate group. So there will be a galactose 1-phosphate, and it will be a phosphorylated, like this enzyme, this substrate, it's phosphorylated. So they will trap the phosphate inside them, and this will cause decrease in the free phosphate number that will inhibit the glycogen breakdown, resulting in hypoglycemia. This is related to the first symptom, which is failure to thrive in the case of the classic galactosemia. For the second one, we have energy crisis in the cell. There's no ATP for the liver, for the kidney, for the brain, so they would have intellectual disability. For the third thing, we would have direct hepatotoxicity. For that reason, we would have jaundice, hepatomegaly, cirrhosis, and they would increase susceptibility to the E. coli sepsis. For that reason, in the case of the classic galactosemia, it's more severe because we have an additional toxin here, which is the galactose 1-phosphate, resulting in all of these symptoms. Unlike the galactokinase deficiency, it is a benign and mild condition, and it doesn't have all of these symptoms due to only the galacticol is being accumulated here. So now we have a comparison between these two diseases. So we said the enzyme is deficient in the case of the galactokinase. In that classic galactosemia, galactose 1-phosphate, uridyl transferase. Accumulation galacticol, here we have galactose 1-phosphate and galacticol. Main symptom is cataract, and here systemic illness, liver, kidney, and the brain. And also there's a clinical manifestation, it would be infantile cataract, while in the case here jaundice, hepatomegaly, vomiting, hypoglycemia due to the galactose 1-phosphate accumulation. In the case of severity, here it's mild, here it's severe and potentially fatal, and we can, in the case of the treatment, like it's not necessary in the case of galactokinase deficiency, but we can do dietary galactose restriction. For that reason, it's not strict, like it's we cannot no need for the treatment here, but we can restrict the galactose. In the case of the classic galactosemia, it's strict, so they would have a strict diet of the lactose and galactose, so they should have stopped eating something containing galactose or lactose, because the lactose contain also galactose. So now we have two questions here. So a 2-month-old infant is brought to the pediatrician because his mother noticed that his eyes look white and cloudy. That means that he have a cataract. The infant has no history of vomiting, no jaundice, no lethargy, and development is appropriate for the age. Physical examination, it reveals bilateral cataract, but otherwise is normal. And the lab study shows no evidence of liver dysfunction. So which of the following enzyme best explains the patient or the infant condition? He have only cataract, and there's everything else is normal. So as we said here, as we can see here, there's cataract only and otherwise healthy. So which enzyme is deficient? Here in this situation, it would be the galactokinase. Would be the answer. So let's go to the next question. As we can see here, a 1-week-old newborn, they develop persistent vomiting, poor feeding, jaundice, hepatomegaly, and lab test shows hypoglycemia. So there would be hypoglycemia or failure to thrive, elevated indirect bilirubin, and also already we said here there would be persistent vomiting, poor feeding, jaundice, hepatomegaly, and the infant later developed E. coli sepsis. So that would be which enzyme is deficient. So that would be the galactose 1-phosphate, because we have a uridyl transferase, because the galactose 1-phosphate is going to accumulate and result in the toxicity of it. There's additional points I want just to mention here, which is the lactase. So it's important to remember that the lactase here, it occurs in the brush border of the small intestine, because if the lactase enzyme is deficient, so that would result in a dietary lactose intolerance, and they could be divided into 3 types. There is primary, there's secondary, and congenital lactase deficiency. In the case of the primary, it's age dependent, decline after the childhood, and it's common like in the Asian and African and Native American descent. While in the case of the secondary, which is loss of the intestinal brush border, so this one it would be lost due to gastroenteritis such as rotavirus or autoimmune disease. And the last one, which is the congenital, it's rare and due to a defective gene. But you should remember the primary and the secondary one. The primary due to aging, and secondary due to loss of the intestinal brush border, small intestine, brush border small intestine, it will be lost due to the gastroenteritis. And that's all. I hope that everything is clear, and thank you.